Welcome to the deep dive. Today, we're plunging into the incredible world right inside you, your circulatory system.
Uh-huh.
Specifically, we're looking at the highways and byways,
Right,
your blood vessels.
right. Arteries, veins,
Yeah,
and lymphatics, too, which often get overlooked.
exactly. You shared some really fascinating sources digging into their biology, and well, our mission today is to unpack how they're built, what they do.
And what makes each one unique,
Yeah.
right? How those differences keep you healthy.
Precisely. We'll be drawing from a pretty comprehensive chapter on vessel wall biology. It covers everything from, the big arteries leaving the heart.
Down to the tiniest capillaries and
Yeah.
that whole lymphatic network.
And yeah, this isn't going to be just a dry science lesson. We really want to pull out the surprising facts.
Yeah, why does it matter that they're built differently?
Exactly. And hopefully give you a deeper appreciation for the frankly amazing engineering inside your own body.
There should be some aha moments, I think, connecting structure, flow, even immunity.
Okay, let's dive in. So this trio, arteries, veins, lymphatics, they work together, but they each have a very distinct job description.
You could think of it maybe like this. Arteries are the high-pressure pipes.
Fizzle tough.
Resilient, they handle the force of the heartbeat.
Okay, high pressure.
Veins are more like the return channels. They need to be adaptable, hold a lot of blood sometimes.
The capacitance vessels, right?
Exactly. Holding capacity, often working against gravity too.
Which we'll get to. And the lymphatics.
Lymphatics. They're kind of the unsung heroes. Cleaning up excess fluid, big role in immunity.
Right. Our source material actually has a table, I think it was table 3.1, that lays out some key differences.
Yeah, it does. Things like how much collagen versus elastin they have.
The structural stuff.
Right. And the pressures they operate under, the sheer stress from blood flow, how stretchy they are, compliance.
Oxygen levels, valves, lots of distinctions.
Definitely. Okay, so let's start with the arteries. They've got that big job handling the heart's powerful pump.
Yeah, those large elastic arteries like the aorta, they're pretty incredible. They don't just transmit the pulse, do they? They sort of absorb it.
Exactly. They act like a buffer. They stretch when the heart pumps that systole, and then they recoil.
Ah, so that smooths out the flow downstream, makes it less jerky.
Precisely. It keeps the blood flow more continuous, like a pressure dampener for the whole system. It stores some of that pressure energy.
And interestingly, the source mentioned that actual changes in vessel diameter, the vasomotor changes, are mainly controlled further down the line.
Yeah, mostly by the downstream resistance vessels, not so much these big elastic ones. They handle the pulse wave. The smaller ones fine-tune the flow.
Okay, that makes sense. And what about blood pressure as it moves through? Does it just gradually decrease?
Well, it decreases, but not evenly. The really significant pressure drop happens in the terminal arterioles.
The really small arteries,
Yeah.
just before the capillaries.
Exactly. And here's something really key.
Yeah.
In those tiny vessels, smaller than maybe a hair's width, like under 60 micrometers, the pressure is actually controlled locally, right there in the tissue. It's largely independent of your main arterial pressure.
Wow. So like each little neighborhood controls its own water pressure.
That's a great analogy.
Yeah,
Yeah, because different tissues need different things at different times.
right. So moving to those smaller muscular arteries, this is where the main resistance control happens.
Yes. They have multiple layers of smooth muscle cells wrapped around them.
So they can squeeze or relax more actively.
Precisely. And how much they can stretch or squeeze depends on, well, the mix of proteins, collagen, elastin, how those muscle cells are arranged, and, whether they're activated or relaxed.
And then even smaller, the arterioles. You call them the traffic controllers.
Yeah, that's really their job. They are the main source of vascular resistance, controlling where blood flow goes.
And they're wired up to the nervous system.
Oh, yeah. Both sympathetic fight or flight and parasympathetic rest and digest. This allows for really specific adjustments.
The source had that example about stress, right?
It did. During emotional stress, the arterioles in your muscles might widen.
Get ready to run.
Exactly. While the ones in your gut or kidneys might narrow down, it's all about prioritizing blood flow for the situation.
Okay, super dynamic. So after the arterioles, blood flows into the capillaries, the exchange zones.
The real workhorses, yeah.
Yeah.
This is where oxygen, nutrients, waste products,
Yeah.
all that exchange happens between blood and tissues.
And it's kind of counterintuitive, but the total volume of blood in all your capillaries is actually more than in the giant aorta.
It is. It's spread out across this massive network. And that huge cross-sectional area means the flow slows right down.
And the pressure drops.
Right. Which is perfect. You need that slow flow and low pressure to give time for all that crucial exchange.
Makes sense. And the lining of these capillaries, the endothelium, It's not all the same?
Not at all. It varies a lot depending on the location and the job. There's a figure, 3.2, that shows this well.
Okay.
You've got continuous endothelium, like in the brain. Cells tightly locked together, that's your blood-brain barrier. Very solutive.
Keeps the brain safe.
Then there's fenestrated, means it has little windows or pores, found in glands, intestines, places where you need more fluid and small molecule movement.
Okay, slightly leakier.
A bit. And then there's discontinuous endothelium, like in the liver or spleen. It has larger gaps, allows even bigger molecules, even cells, to pass through.
So the structure is really tailored to the specific exchange needs of that organ.
Absolutely. Form follows function, perfectly illustrated there.
Okay, so blood leaves the capillaries and enters the venules, the start of the return trip.
Yep, the post-capillary venules first, then collecting venules. They're kind of the mirror image of arterioles, but on the low pressure side.
Thinner walls, slower flow.
Thinner endothelium, yeah. And they are quite leaky, actually. Slow flow, low pressure.
And that leakiness is important for inflammation, white blood cells getting out.
Exactly. It allows white blood cells to squeeze through the gaps between endothelial cells, that's called diapedesis, to get to sites of infection or injury.
Fascinating. Okay, venules merge into veins, and veins are known for being stretchy, compliant.
Very compliant. That's their key feature. They can handle big changes in blood volume without much increase in pressure.
How do they do that?
Well, the source mentions their shape can actually shift. When they're relaxed, they might be kind of flattened or elliptical. As they fill, they become more circular.
Ah, and a circle holds more volume for the same perimeter.
Exactly. Significantly increases capacity. A relaxed vein might double its volume with just a tiny pressure bump.
Wow. And their outer layer, the adventitia, is strong.
It's rich in collagen, provides that structural support so they don't overstretch.
But the real stars of the venous system, especially against gravity, are the valves, right?
Absolutely crucial. Those one-way doors, they prevent blood from just pooling in your feet when you stand up.
Where do you find them?
Most veins bigger than about 2 millimeters have them, usually just downstream or distal, to where a smaller vein joins a larger one.
And they're made of
A core of collagen and elastin, covered by that endothelial lining. Strong but flexible flaps.
this source mentioned an exception in the feet.
Yeah, bit of a unique setup there, helping shunt blood from deep muscles towards the surface veins. It's all about getting blood moving upwards.
And when these valves fail, that leads to problems.
Big problems. That's where things like chronic venous insufficiency and varicose veins come in.
Right, the bulging veins.
Yeah. If the valves are leaky, blood flows backward, pressure builds up in the lower veins.
It causes swelling, pain.
And over time, the source says you can actually lose valves and the remaining ones can get damaged. There's inflammation, remodeling of the tissue. It's a downward spiral driven by that high pressure.
Yikes. Okay, and the biggest veins, the venae cavae.
Yep, returning blood right to the heart. They're huge. They even have their own tiny blood vessels, the vasa vasorum, to nourish their thick walls.
And some heart muscle tissue near the heart.
A little bit, yeah, in the outer adventitial layer right as they join the atrium.
Okay, let's switch gears slightly. The lymphatic system, running in parallel.
Kind of in parallel, yeah. But it's a one-way street, not a circuit like blood. Its main jobs are fluid balance and immune function.
The body's drainage and filtration system, you called it?
Pretty much sums it up. It starts with these tiny dead-end vessels, initial lymphatics.
Where are they?
In the interstitial spaces, the gaps between your cells and tissues. Everywhere, really, but especially dense in the skin and linings of organs.
The source mentioned the lower limbs have a denser network to fight gravity.
Seems likely, yeah. A greater capacity to transport fluid, probably to help counteract that gravitational pooling.
And how does fluid actually get into these initial lymphatics? It sounds tricky if they're dead ends.
It's really clever. There's a figure, 3.3, showing these microvalves. The endothelial cells lining the vessel overlap like loose flaps.
Okay.
And these flaps are anchored to the surrounding tissue by tiny filaments. When the fluid pressure outside the lymphatic increases.
Like when tissues swell a bit.
Exactly. It pulls on those filaments, which pulls the overlapping edges of the cells apart, creating little gaps. Fluid flows in.
And when the pressure drops.
The flaps close, so fluid can get in, but it can't easily get back out. It ensures one-way flow right from the start.
Ingenious. So where does the fluid go from there?
Into pre-collecting lymphatics. These start to look a bit more like vessels. They develop secondary valves inside.
Like the veins.
Similar, yeah, but maybe spaced more irregularly at first, sometimes just single flaps. and smooth muscle starts to appear around them.
Ah, so they can start to push the lymph along.
Exactly. They have a dual role, still absorbing some fluid, but also beginning active propulsion.
And then the collecting lymphatics.
These are the main transport vessels. They have a more complete structure, a proper basal lamina supporting the endothelium, more regular and developed valves inside.
And more smooth muscle.
Yep, a layer of smooth muscle, often arranged helically, within the vessel wall itself.
And this is where lymphangians come in.
Right. A lymphangian is just the segment of a collecting lymphatic between two valves.
Okay.
And these segments act like little pumps. They're innervated, they contract rhythmically, spontaneously, squeezing the lymph progressively along the network.
Like a tiny chain of hearts pushing the lymph fluid.
That's a great way to visualize it.
Okay, so arteries, veins, lymphatics, very different jobs, different structures. But they share a basic wall plan, right?
They do. That fundamental three-layer structure. The inner intima, middle media, and outer adventitia.
Though the source notes it's less clear-cut in lymphatics.
Right, the layers aren't as distinctly defined there. And another key difference, arteries have those prominent elastic layers, the internal and external elastic laminae.
Which you don't see as clearly in veins.
Much less prominent, yeah. Figure 3.4 gives a nice visual comparison side by side.
Now, beyond the physical structure, you mentioned molecular identities, like cell signatures.
Exactly. This is fascinating. The cells themselves know what kind of vessel they belong to. Endothelial cells lining arteries express a marker called Ephrin B2.
Okay, arterial marker.
Venous endothelial cells express FB4, and lymphatic endothelial cells express PROX1.
Different flags for different teams?
Precisely. And there are others, like VEGFR3, specifically in lymphatics. Table 3.2 lists quite a few. It's how these vessels establish and maintain their distinct characters.
And this isn't just the lining cells, even the smooth muscle.
Even the smooth muscle cells can have unique molecular fingerprints depending on where they are.
The source had that amazing example about the aorta.
Oh yeah, the thoracic aorta versus the abdominal aorta. The chest part is much more resistant to atherosclerosis, plaque buildup.
And that's linked to gene expression, HOX genes.
Exactly. Differences in HOX gene expression in their smooth muscle cells seem to program this difference in susceptibility.
And even if you swap them surgically in an animal,
they retain their original fate. It shows how deeply ingrained these cellular identities are.
Wow. Okay, well, you mentioned FMB2 and FD4. How do they establish artery versus vein identity?
They're part of a critical signaling system. FMB2 is on arterial cells, FAB4 on venous cells. When these cells touch during development, they signal to each other. It's bidirectional signaling. This interaction helps sort them out, ensuring arteries connect to arteries, veins to veins, and setting up the boundary.
So it's like cellular communication defining the blueprint.
Absolutely. And disrupting this signaling causes major problems in vessel formation.
What happens downstream of that arterial signal, FMB2?
In arteries, it involves activating other pathways involving molecules like VEGFR2, DLL4, Notch. These help reinforce the arterial identity.
Okay.
And importantly, they also suppress a protein called CoTTFII.
And a CoTTFII wants to make veins.
Exactly. CoTTFII is a key driver of venous identity, so arteries actively suppress it.
Interesting. And FRNB2 also plays a role in inflammation later on.
Yeah, in adult arteries, it can interact with immune cells, monocytes, potentially influencing how they behave during inflammation.
Okay, so on the flip side, COPTFII drives the venous fate.
Yes, it's a master switch, a transcription factor. It turns on genes like AFB4, the venous marker, and actively suppresses the arterial program.
But you mentioned something surprising about vein grafts losing AFB4.
Yeah, kind of counterintuitively, when a vein is used as a graft in the high-pressure arterial system, like in bypass surgery, losing that AFB4 signal seems to be important for it to adapt and mature properly in its new environment.
And lymphatics, what's the key player there?
That's PROX1. It's essential for lymphatic development, turning venous endothelial cells into lymphatic ones. And its expression is controlled by factors like COPTFII again, and SOX18, and VEGFR3 signaling. It's a whole network of molecular signals.
A complex dance indeed. Okay, last major topic, hemodynamics, the physical forces.
Can't talk about blood vessels without talking about forces. Two main ones, cyclic strain and shear stress.
Cyclic strain is the stretching from the pulse pressure.
Right. The vessel wall stretching and relaxing with each heartbeat. And shear stress is the frictional force.
Well, the blood flowing against the wall, like water in a pipe.
Exactly. And these forces aren't just passive. They actively shape the vessels.
How so?
Well, shear stress mainly affects the endothelial cells, the lining. It actually makes them align themselves in the direction of flow,
Streamlining.
pretty much. And interestingly, higher shear stress in arteries makes these cells build up more internal scaffolding stress fibers. But the same stress level in veins doesn't trigger that response.
So different responses even to the same force.
Yeah. Shear stress also triggers the release of vasoactive stuff like nitric oxide, NOO, and PGI2.
Things that make the vessel relax and widen, vasodilate.
Correct. And long-term, high shear can boost the whole NO system, activate remodeling enzymes called MMPs, the vessel adapts.
And low shear.
Low shear can actually hinder vessel growth or cause vessels to regress. Flow matters.
Okay, and the cyclic strain, the stretching.
That affects all the cell types in the wall.
Mm.
Smooth muscle cells tend to orient themselves perpendicular to the stretch.
Interesting, at right angles.
Yeah. And the amount of stretch influences their behavior, too. They might grow or enlarge or change the proteins they make.
So more stress means?
Generally, increased circumferential stress, the tension around the wall leads to smooth muscle cells getting bigger, hypertrophy, and making more collagen and elastin. Making the wall thicker or stronger
And less stress.
leads to atrophy, cells shrinking. The vessel tries to adapt its diameter and wall thickness to maintain a normal level of stress.
Which explains why chronic high blood pressure leads to thickened artery walls.
Exactly. The vessel remodels in response to that sustained high stress.
And these forces don't just change structure, they influence identity, too.
They absolutely do, both during development and in adults. The source mentioned blocking flow in a developing artery.
It loses its arterial markers.
Yeah, and putting a vein in an artery spot.
It starts to lose its venous identity. The environment overrides the original program to some extent.
It shows how dynamic vessel identity can be. There was also that example of veins near an arteriovenous fistula.
A direct connection between an artery and vein.
Right. Those veins experience both higher shear stress and more stretch, and they can actually start expressing both arterial and venous markers, a kind of dual identity.
Wow. So the forces are constantly talking to the cells.
Constantly. Figure 3.7 shows how those forces get transmitted from the outside through receptors into the cell, changing gene expression. Physical force becomes biology.
Incredible. So wrapping this up, what's the big picture takeaway for us?
I think it's that your circulatory system isn't just plumbing. It's this incredibly dynamic, specialized, and responsive network.
Arteries built for pressure, veins for volume and return, lymphatics for cleanup and immunity, all distinct.
With unique structures, unique molecular signatures, all shaped by the job they do and the forces they experience. They're constantly adapting.
It really gives you an appreciation for the complexity.
Definitely. And maybe an intriguing thought to leave with. Consider how even a subtle change, like blood flow patterns, can trigger this cascade of biological events, actually altering cell identity.
Yeah. Makes you wonder what other subtle forces or environmental things in our daily lives might be influencing this microscopic world inside us.
Exactly. And could understanding these mechanisms better lead to new ways to stay healthy or treat diseases like hypertension or varicose veins or even problems with lymphatic drainage?
It feels like we've only scratched the surface, really.
Oh, absolutely.
Wow.
Vascular biology is a huge and fascinating field. There's always more to explore.